There we go. Okay. So I'm hoping that you have seen, so again, just for those of you who weren't here right at the very beginning, I do have two screens going. So if you see me looking over to the side, it's not that I'm ignoring you. The slides that I'm going to show you can be found at bit.ly/biocdl unit 4 webinar. I'm going to put that into the chat so that you can access that more easily. There are some links there that might be helpful for you. And we have used the same setup for all of the other webinars that we've done in this series. So you can just replace the unit number in that URL and it will get you to the slides that are relevant to that webinar for that particular unit. And what we've also done here, I'm on slide number 5 if you're following along in those. And what we've done is that we've linked here the traditional unit 4 outline, which we would have used in previous years, and then the distance learning unit outline, which is the newly revised one, so that you can compare and contrast. The traditional unit outline would not be one that would have been edited at all, so you can see what we would have done in previous years. This unit is quite, we've had to shift a lot of things around to get students to have a little bit more agency. So for all of you, I think, almost all of you anyway, have been to a summer class around patterns biology, either with me or with Caitlin Everett or in coordination with Jesse Chamberlain. And so you're probably familiar with the principles, the design principles around the patterns approach. We're not going to go into those in detail today, but there's a link, there are 2 links there to the short version and the longer version about the ideas about why we pick some of the types of activities that we pick. And we'll have a chance to take a look at one of those at the end of today. So the unit 4 essential question in CDL has been shifted a little bit in the past. If you're familiar with this unit, you may remember that we had a essential question that was focused on breast cancer and diabetes. But in order to, as I mentioned before, give students a little bit more agency and relevance for them, we've actually expanded the idea here to be more about just disease in general. That actually plays out at the end of the unit where they're able to write their project about any disease or condition that may interest them. And so we've kind of backed away from the specifics of those 2 diseases, breast cancer and diabetes. So what you're going to be seeing here is how we've tried to make some things a little bit more general, but while delving in a little bit more deeply into breast cancer, or excuse me, into cancer generally, as an option or as one example, I guess. So the learning target is basically the same, right? Looking at how genes and environment interact to determine traits in populations. And again, the anchoring phenomenon has been kept basically the same to indicate that there are disparities in how people in different groups and in different geographic locations get disease, and particularly diabetes and cancer. So we've kept that anchoring phenomenon the same, but the idea is to try and expand that, use diabetes and cancer as an example for things that are happening in a variety of diseases and conditions. So what I'm going to be doing here is going to be highlighting, going through the task sets. I encourage you to ask questions. We're a small enough group that you can certainly unmute yourself if you have a question on a particular task set. Some things are the same as they have been in the non-CDL version, and some things have changed or been enhanced, and we've tried to identify those enhancements or changes on the slides and to draw particular attention to those, because if you're familiar with the unit as it was before, you'll want to know what's different in CDL. So the engagement task set, the unit opener here, is actually almost identical as it has been in previous versions of this unit. Students are introduced to some maps and graphs that are collected from the American Cancer Society and the CDC and they reflect on what they're observing in those maps. And those maps and graphs indicate geographic distribution, racial and ethnic group distribution of cancer diagnoses, cancer deaths, diabetes, and diabetes diagnoses. And so it's just an opportunity for students to take a look at some data and to ask questions. And so in the INB, in the interactive notebook for this unit, there is a built-in reflection task option or task document. It is also available as a separate standalone document if you're not using the INB. But this is not different from what we've had in previous versions of the curriculum. And there is a tracker for students to use. I'll point it out at the end when we look at some additional resources for you. But if you've been following along with what we've been doing over the course of the year, that tracker is just a way for students to, at the end of every task set, identify how they're getting closer and closer to answering the big unit question based on the arc of learning during that particular component, or that component of the arc of learning. All right, so if you did unit 3, or maybe you're working on unit 3 right now, you may have, I'm sure you noticed that unit 3 was dramatically pulled back to really focus on feedback loops and the example of carbon dioxide and oxygen concentration in the blood as a homeostatic situation and the mechanism of keeping that homeostasis. So because of that, there were a couple of learning targets and NGSS standards that we removed, or we decided to remove from unit 3 to keep that really tight around that homeostasis phenomenon. And as a result, the learning target around cell division and cancer was removed from unit 3. And we thought that there was a variety of reasons for that decision, but it made sense to us that since cancer is part of the initial question asking task set, that we describe here in this unit what cancer is and how it happens. So the actual task here around cancer is not changed from what it had been in previous versions of unit 3. It's just been placed now in unit 4 to follow up those data around cancer. On the other hand, the mitosis lab, looking under the microscope, looking at the phases of mitosis component, has been removed. Those things are still available in the original unit 3. If you want to go back to those, there are options for using some online. There are some online options for mitosis, but the NGSS standard is very clear that the steps of mitosis are not a testable topic. And we felt that the relevance of cancer and how cancer actually develops was more important for us to focus on rather than the steps of mitosis. I included a couple of videos that do cover very basically the ideas of how the chromosomes separate during mitosis, but just to be aware that that is not a focus in this task set. So this understanding cancer activity comes from an old lesson from NIH and discusses some factors that cause cancer, such as exposure to chemicals, ultraviolet light, other kinds of radiation like X-rays, and also that genetics can come into play. The idea there is that that's kind of foreshadowing what's going to be coming next in this unit around epigenetics and, of course, how some kinds of conditions and diseases can be inherited. There is a CER assessment prompt in this task. It's the first assessment in this unit. And it just asking students to be able to reiterate in their own words, what are some things that can cause cancer and how cellularly does cancer occur? So the idea about mutations can happen to certain genes, which are called tumor suppressor genes and proto-oncogenes, and then those can cause changes in the cell cycle. Again, if you have questions, feel free to unmute yourself. for the moment, these are things that have been just either kept the same or moved to a new location in the curriculum.
I'm gonna put, we did that and we just reformatted it a little bit easier to put kids in breakout rooms. I'm gonna put a link in the chat so you guys have it. It's just
great. Thank you so much. Yeah, doing with breakout rooms, I've had depending on the day, almost like different levels of success with breakout rooms. It's really hard. I'm trying to balance, but thanks, Heather. I'd appreciate anything that you can share. We can always put that in as a separate, as an alternate option for formatting. Thanks. So moving on into the exploration, this is where we're starting to get into some things more specifically about why, getting to the heart of the essential question here. And the main part of, or the main standard in this case, is around variation. And so again, the NGSS performance expectation and assessment boundary are very clear that the biochemistry and things that happen on a very specific level in the cell are not part of the testable topics for meiosis. And so we're really taking the idea of variation at kind of a umbrella or bird's eye view, like what are the ways that variation can happen? And so this task has been revised for CDL. It's a very student independent, hopefully, task. It's an alternate setup of alternating videos and questions to allow students to investigate the different kinds of ways that variation can occur. So focus, of course, on meiosis, random fertilization, mutation, so that students have and the standard very clearly says that there are several different ways that variation can occur. And that's what we tried to focus on, was looking at the standard and making sure that students had the opportunity to see all those variety of ways for variation. This activity or series of activities can happen either in the IMB or as a standalone, depending on whether you're using the docs version of the IMB. And then lastly, in this task set, we're giving students an opportunity to begin to collect some data from family members in an anonymous way, giving them an option to use a Google form. Now, we've traditionally done this task after the holidays, and so we've given students an opportunity here to be able to communicate with their family that they might not be seeing, or very likely are not seeing. And we've made all of the questions in the Google form optional, so that family members can choose to just report what kind of conditions or diseases that they are, or epigenetic factors that may be influencing their lives, and not given their name. You may have noticed that the pedigree project has been removed from the unit. That would normally be, we'd normally be introducing that very early on. And so it's just one way for students to collect information. We're not expecting that every single student would do it this way, but it's a way for them to get some information about conditions or diseases that may exist in their family, so that they can kind of gather some information about what type of condition or disease they might want to focus on for their final project. Task set 4 is a shortened version of Punnett Square task set that we've used in previous years. When we looked through the NGSS standards that would be relevant to this unit, we felt that if you were short on time, that this task set might be one that you could consider skipping. Now, I know that might cause some questions, or cause some questions, because the idea here is, we know that, in my experience, I should say, my experience is that students, when they come across Punnett squares, they feel, and they're able to master them, they come to a sense of pride because they remember how to do some of those things from their time in middle school. And if they don't remember, they can relearn to do those with, there's a certain process, right? An algorithm that they can follow to do Punnett squares. On the other hand, as a biologist, I know that there are so few human traits that are actually controlled in this way that that is where my hesitation is to spend a lot of time on this particular task set, because I don't want students to come across the idea or to think that most or many human traits are in fact controlled in a Mendelian fashion. So this task set does include codominance and codominance in terms of blood type, multiple alleles, of course, and then also sex linkage. And we used all human traits that have been demonstrated to be passed in those particular fashions. So if you're running short for time, we feel that this might be a task set that you could skip. But I also recognize that, you know, this may be a level of comfort for you and for your students. And so I was able to do this in one day. I introduced one example of each type, and then I linked some videos in for students to be able to refer back to for help on solving the problems in the problem set. But I also really focused on, when I was doing this task set, also really focused on the idea that many human traits are not covered in Mendelian fashion.
Charlotte?
Yeah, Christine.
Sorry, when you said you did this in a day, did you do it in a 40-minute class day or a 90-minute?
So we're in the recommendation in our district is that we have 70-minute periods, but we're not supposed to be live with them for more than like 35 or 40 minutes.
Okay.
So what I did is that I did in that 35 minutes, I did one example live. I had my iPad and I actually drew the Punnett squares out. I did different problems from the ones that are in the problem set. But then in the problem set, we've also linked in some videos from Amoeba Sisters and other resources for students to be able to see other examples and some other background information about those different kinds of inheritance. And then they had the problem set to do.
Okay, thank you.
Secret, that's a good question. They are in the practice in the problems, and there is a slide deck, and it's linked in I forgot to link it on this slide, but it is in the unit planner, which my little I don't know if you guys can see that, but up in the top right corner of the slides, the distance learning plan is linked there, and the slide deck is already linked in this planner, and it's linked in the folders too. So.
Charlotte, I have a quick question.
Yeah.
Do you see students 4 days a week with that 70-minute schedule?
Yes, we do.
Okay.
Yeah.
Thanks. Just trying to calibrate what we can possibly do given our schedule.
Yeah, we're seeing students Monday, Tuesday, Thursday, Friday, and then Wednesday. We're supposed to provide asynchronous work for students to do on Wednesdays, but what we're seeing district-wide is a very mixed bag in terms of whether students are doing any asynchronous work on Wednesdays, and how much, if they are doing some, how much are they doing? So I'm, for the most part, not ever introducing anything new or completely independent for students to do on Wednesdays. I'm usually trying to plan it so that like we do something Monday, Tuesday, and then they finish it on Wednesday. That's kind of how I've been approaching it. So.
Okay, thanks.
So thanks, Kyle. I saw your note there about the Blue People of Kentucky. Yeah, they can do that particular. Yeah, I've read that case study in the past, and I've not ever integrated in. So again, I'd love to see if you can share that over. We can put that in as a extension or alternate. Yeah. Sorry, my 16-year-old did not realize that we were time out now. Okay. moving on. So task set 5, and this is where there's been some significant addition for this unit. So task set 5 is where we're doing epigenetics, and this is a longer task set. The first part, if you're familiar with this unit, is something that you've seen before, and even if you're not familiar with this unit, you may be familiar with the Learn.Genetics from the University of Utah. They have a really great lesson around epigenetics with the Lick Your Rats activity. So students are able to actually see how the nurturing of a mother rat actually can change the coiling of the DNA in their babies, and that uncoiling and subsequent demethylation can actually be passed on to future generations. And so it affects actually how those rats are, how they handle stress. And so in the past, we've used that activity as the sort of the main example of how the environment can affect the genome, and therefore this idea of epigenetics or epigenomics. The addition here has been to include now some information and a whole second part of this task around intergenerational trauma. So this is obviously a very sensitive subject. And we worked, I will say, I did not work specifically on the development of this task set, Jesse Chamberlain, and we had a group of people on the Biology Council from Hillsborough and Portland Public Schools who worked with this also, and they worked in conjunction with, we have a social worker, Tosa, in Beaverton, who helped that team develop and curate some resources for this part of the task set. So there, and it incorporates some very recent news articles and reports that I heard on OPB about how certain populations, or really everybody, is experiencing stress right now during the pandemic, and how that might exacerbate some previous situations of trauma that families or people have experienced. And so trying to incorporate those plus other articles and activities around intergenerational trauma. So there are lots of resources in the teacher and student materials for you as teachers to look at and use to prepare yourself for incorporating this lesson into your classroom. I encourage you to look at them. This is a lesson that I know I'm going to have to spend a few hours to prepare myself for how to incorporate it especially into my online class. So, just a suggestion that you may want to read the articles in advance, and you may want to have a conversation, if you have an opportunity to have a conversation with a school counselor about if you're apprehensive about how to incorporate some of this material. But there have been studies that have shown that these kinds of events over long periods of time can actually have an effect on the epigenome. And so trying to give a little bit of relevance, but we understand that there can be there's sensitivities around this topic, especially right now during the pandemic. But we wanted to give students an opportunity to learn about that. And one reason for that is because one of the options that they may choose to investigate at the end of this unit for their project would be the effects of that for anxiety, depression, or other kinds of conditions that they may be experiencing or have seen in their family. So something definitely to look at. Speaking of the project, we've built in a task set here where students are able to take some time to parse through the data that they may have been receiving from their families about conditions or diseases that exist in their family, or for them to do research about any other disease or condition that they're interested in looking at for their final project. So what we have done I'm going to go ahead and actually click on this link. And I'm going to share a little bit differently so we can see it a little bit more detailed. There we go. Make sure this is a little bit larger so we can see it better. So the project at the end of the unit is for students to make some kind of poster or video or brochure for a community of their choice. And so the way that this has been modified sorry, let me make this zoomed in some. There we go. The way that we have modified this for CDL is that students are able to choose, as I mentioned, beyond just breast cancer and diabetes. We have actually already generated a whole list of resources around a bunch of different conditions. So we have a resource sheet for, of course, for diabetes and for breast cancer, but also now for alcoholism, skin cancer, melanoma, Parkinson's disease, colorectal cancer, prostate cancer, and Alzheimer's disease is coming. That one will be finished up in the next week or so. So students can choose from any one of these that have the resources already identified for them, or they can choose any kind of any condition or disease that may exist in their family. We have, I will say that if you have a particular condition or disease that you think would be helpful to add to this list, we had a limited number of people who were able to participate in the collection of these resources. But if you or your students come up with a list of resources for any other condition or disease, we would love to add it to this list. And you can see that if we click on, let's say the one for Parkinson's disease, we have a template, and that template includes all different kinds of podcasts and articles and videos about Parkinson's disease, specifically around different kinds of groups that might be interested in information about that. And then also the research that that links genetics and genomics and the epigenomics to that particular disease. And then we've provided also some graphic organizers for students to be able to collect some information. So Unit 4 Task Set 6 is really an opportunity for students to, let me actually get this back here now, my smaller version. Oh, just get this out of the way. I'm using this new feature on Zoom where you can share a part of your screen. So in Task Set 6, they have an opportunity to actually do some research about, well, okay, which kind of disease would I be interested in investigating? And they can look at the resources that we've already curated for them, or they can go out on their own and do some research on a different topic. So that's kind of a day in there that you can kind of play around with, but to give some students some agency around which topic they're going to pick for their project. All right, I'm curious how many of you are familiar with the SB 13 Tribal History Shared History obligation. You can just either give me a thumbs up either on your screen or in Zoom on the participants window if you're familiar with that. Good, so some thumbs up there. So SB 13 was passed, I think, 2 years ago, and the Department of Education worked with tribes in Oregon to develop lessons, and these are actually now required lessons. I think that they're at grades 4, 7, and high school. They're mostly written for grade 10, but districts have the ability to incorporate them anywhere in the high school curriculum where they see that that's appropriate. And there are lessons that are written for ELA, social studies, and science at a minimum. There may be more than that. But last year, the expectation, we're supposed to, and I say we, district are supposed to incorporate these lessons into the curriculum last year, but there was some relaxation around those restrictions as a result of schools being closed. This year, it's been stated very clearly that the expectation is that we, as districts, incorporate these lessons this year. There are 3 science lessons that have been booked. 2 of them go into the from our perspective, fit best into the biochemistry curriculum. So this one is one of them. There's one about that fits more into Unit 1, but we will. That lesson actually was published after Unit 1 was already taught this year, so we will be incorporating that into Unit 1 for second semester. So those of you who are in Hillsborough who are repeating Unit 1 right now, you'll just do it next year, I guess. But just be aware that if you're not hearing about these from your district, that there's a reason why this lesson is in here, because of the tribal history, shared history, Senate Bill 13 that has been passed and is now expected to be implemented statewide. So the lesson here is about food sovereignty effort, different tribes, and the way we have taken our spin on it to put into this unit is to make that connection with the higher rates of diabetes that appear in Native populations. And so there's an activity guide, which has mostly come from the tribal history, shared history curriculum. And then we have added something to that to make a more specific connection to the rest of the content in this unit. So here, the SB 13 ODE website, which gives you access to all of the lessons that have been developed for all of the subject areas and all of the grade levels. And then so helpful, we do have a Native tribal member on our team who helped with the redevelopment of this lesson, or the modification of this lesson to fit into this unit. And she found these 2 resources, which she thinks would be helpful for non-Native members to implement this lesson. So we did have this in the last year, but I personally skipped it for lack of time when the restrictions about implementing it were relaxed in the spring. And then the last topic in this unit is the project. So the project itself has not changed, except to give students a little bit more flexibility about what they choose. So the idea behind this project is that students are going to be making some kind of product to communicate with a community of their choice about a topic, disease or condition of their choice. And the idea would be them to make some kind of tour or poster or video to communicate relevant genetic, genomic, and epigenetic information about that particular disease or condition to a community that is potentially at higher risk for that condition or disease. So in the past, I've had students who are Native students who have to write their project for members of their Native population. I've had students who come from who have heritage in other countries to write a project for their community. I have encouraged students to write the project in their native language, if that is relevant to the target audience that they select. So this is very open for students to be able to select something that's really relevant. So I gave you a quick view a little bit earlier of the documentation that students have for that and the resources that we have developed to provide them some basis for this research. Again, they're again welcome to make a selection of different of a different topic if you wish. So let me give an example. Just quickly run. I should have linked this in. Example project that I just want to show you. So here's a brochure that was done in a previous year about diabetes, and in particular, the target audience for parents of young children. It's taking it to load here, sorry. This was created with Canva. You have to imagine what it would look like if it were in a trifold or folded setup. So this one is written for kids and diabetes, what you need to know, and it incorporates information about statistics and questions, and some information about what they might need to know, graphs, recommendations that that families and children can take about what to do if they are experiencing prediabetes or have been diagnosed with diabetes. So this example is linked in the unit planner. But students can do any kind of topic that is relevant to them. Certainly it would be a little more challenging for them to do that. We have not curated resources for, but again, if you have a topic that you think would be of particular interest and are able to gather some resources, that we would love to take that information and incorporate it so that others can benefit from the time that you have spent doing that research. All right, so in terms of design principles, what we've tried to do in thinking about the revamping of this unit is to try to make it student-centered. Just like you should take a minute or so and look at this diagram about the next generation model of science education, science instruction, and think about this question, and if you could write an answer in the chat. How does the next gen model that's just demonstrated in the picture allow teachers to leverage student assets and cultivate STEM identity, which are both of those are components of student-centered learning in the PAS approach? How can we do that in light of CDL? So just take a minute to take a look at that diagram and write an answer in the chat. thank you to those of you who've given us a little bit to think about. Give another 30 seconds or so for people to finish up their thoughts, and then we'll read those out. That's great. What I'm seeing in your messages in the chat is that students have a lot more agency. They have more choice here, right? It allows them to be more, potentially be more engaged in this situation where we are physically separated from each other, and therefore makes it more difficult. Well, I mean, I know I've definitely experienced challenges and engagement. So what we have been trying to do in the revamp of this unit to get any possible way for students to be able to make meaning. I think somebody used that terminology, crafting meaning. Kelly, you said that. Trying to release, like you're saying, Secret, release the responsibility completely from the teacher, right? Because we know we have so many other things that we're trying to handle right now, and to give students an opportunity to take more responsibility and to have more, make that connection. And Kyle said to make that learning more manageable, right? For them, if they're able to make those choices and what it is that they're learning, then they can, then they feel more connected to that. Right? Those personal experiences and family history. Right? Thank you very much for those comments. Good. Yes, Crystal, I agree. Right. We know that the level of autonomy is challenging for students. We talked a little bit about that already, but we hope that this personal connection gives students a little bit more connection. Let's see, just a few more things. I don't think I mentioned all of these. I think I did mention all of them. So there are 3 possible summative assessments as we see it in this unit: the CER around cancer, another CER writing task around epigenetics, and then the community education project at the end.
Charlotte, how
Yes.
long? Sorry, how long does this go on?
So I'm right in the middle of it right now. I'm about to start task set 4 on Monday, and I'm expecting it to have. I'm really hoping for 3 weeks on our 4-day, meeting with students 4 times a week schedule.
You mean 3 weeks from the time you started, or 3 weeks from now until the end?
3 weeks from the time I started.
Okay.
I'm really shooting for 3 weeks per unit to make sure that we can get through to unit 6 by the end of the semester.
Okay. All right. Thank you very much.
We have 18 weeks. We knew that. That's one of the reasons that we actually shortened unit 3, was because we knew that unit 1 was really long,
Huge.
even though we pulled it way back. So that's one reason why our classes, Secret, are we have 70 minutes, but we're not supposed to be live with students for more than 35 or more minutes per day. So it's supposed to be like live instruction for 30 to 40 minutes or so, and then 30 minutes of asynchronous work. But we are allowed to do is that, like, work time in a small group, like, or a breakout room, does not count against our live time, even if it's during that 70-minute Zoom period. So the part that I would be, I'd like to say I lecture, but the part that I would need to be recording shouldn't be more than 35, 40 minutes. So I hope that answers your question, secret. So we've also tried to move away in the unit planner from using days in the calendaring, trying to move towards minutes, and trying to blanket or to separate it between sort of live time and asynchronous time, so that it's a little bit clearer for teachers who might be on a different schedule. So that's a good question about Flash. So it doesn't go away in December. It's just not going to be supported anymore. I have a message into University of Utah to find out what their plans are. I think that they're really hurting for money right now. They know that Flash is problematic or going to be problematic here very quickly. What we have experienced is that students are still able at this point to run the animations. The real problem that we're having is any videos that are on the University of Utah site are run through Vimeo, and we are not having success with students being able to see those when they're district devices. They are able to run the Flash extension, at least now. My Mac keeps telling me that Flash will no longer be supported, but I don't know what that means yet. I don't think we'll know until December 30th. Not sure whether those things will just stop working altogether or not. So right now we're working with what we have. We won't know what the implications of that change will be until it happens. It is 11, so I do want to respect your time. So if you have other agendas I hope you have something else, other agendas to work on and things to work on your Saturday morning into the rest of your Thanksgiving weekend. I will hang out a little bit longer if anybody has additional questions. I just want to point out to you that there are some other supports for Unit 4 on the top of the unit planner. I think all of you have access to the teachers only folder, but if in case you don't, there is a link there for you to fill out a form, and we will get you time or access to that teachers only folder. I think that might only apply to people who are brand new to patterns or who are student teachers. So I'll hang out for a little while longer. Thank you guys for coming, and have a happy rest of your Thanksgiving weekend.
Thank you so much. I appreciate you.
You're welcome.